Hey guys, my bad. I started the wrong meeting. Well, what questions do you guys have? You guys on?
Yeah.
All right. What questions do you have?
I think it would be helpful if we could go back over the different types of like photosynthesis and all those sorts of processes.
Okay. So who can tell me what they remember about C3 photosynthesis? So the two forms of photosynthesis, do they differ in the light reactions or in the Calvin cycle?
It's the Calvin cycle, right?
Yeah, so it's the Calvin cycle where they differ. So what are the fundamental differences if you were to like just do a compare and contrast on how C3 photosynthesis is different from C4 photosynthesis?
I literally just wrote that on the study guide.
Ha-ha.
C3 and C4.
I sent Dr. Judd my part of the study guide and he's working on his. Okay, so I'll help you through it. Where does carbon fixation happen in C3 and C4 photosynthesis?
Well, don't they happen like in different places too of cells?
They don't, not for carbon fixation, not for that initial capture of carbon.
Well, I mean between C3 and C4.
Yes, between C3 and C4, the initial capture of carbon occurs where in the plant leaf?
Would that be at the stomata?
No, that's where CO2 enters the leaf so that it can get into the interstitial spaces between the cells of the leaf, in between those mesophyll cells. But where does the actual carbon capture occur? Well, and by carbon capture, I mean the carbon dioxide is taken and added to some other molecule.
Does it start with an M?
It does start with an M, and I just uttered it a minute ago.
Is it mesophyll or something like that?
Yep. So that initial carbon capture happens in the mesophyll cells or the mesophyll cells, however you want to pronounce it. I don't really care. It all happens there. But in those cells, C3 and C4 photosynthesis differs in terms of how the carbon gets captured. How is that different in those mesophyll cells? In C3 photosynthesis, the carbon dioxide is going to be bonded to what molecule?
Almost everybody got this right on the homework. You guys know it.
So the C3 was Rubisco, and then the C4 was at the PEP carboxylase.
Okay, so since that thing is carboxylase, what does that mean that thing is, that ASE ending on the end of it?
An enzyme.
That's an enzyme. So those two enzymes are going to do something. They're going to grab carbon dioxide, and they're going to add carbon dioxide to something else. In C3 plants, what do they add carbon dioxide to? What does Rubisco stand for? And I don't remember if Dr. Judd did this, but I always spell Rubisco with a capital R, lowercase u, capital B, lowercase is, capital C, and capital O, because it helps me remember what that name means. What does the RUBP stand for? Or the RUBIS stand for, rather? Duh, I just gave it away. What does the RUBIS stand for? It's the 5-carbon compound that Rubisco adds carbon dioxide to. What's it called?
Now they're looking.
What's that, Nathan?
I see them looking in their
Oh,
notes.
okay. I can't see anybody looking. The only person I see is Joseph.
Oh,
Video
yeah.
wise.
Now I see.
C3 photosynthesis, what does CO2 get added to by Rubisco?
Is it like the RUBP something?
It is RUBP, and that's all it is. It stands for ribulose bisphosphate. And Rubisco is ribulose bisphosphate carboxylase oxygenase, because Rubisco could either add a carbon dioxide to ribulose bisphosphate, or it could add an oxygen to ribulose bisphosphate. So that's what happens in C3 plants. How does carbon get fixed? What do the plants add carbon dioxide to in C4 plants? Remember, this is done by what enzyme again? You said it earlier. Somebody said it earlier.
The PEP carboxylase.
Yep, PEP carboxylase. And so what compound does PEP carboxylase add carbon dioxide to?
I might not have given him that one.
Oh,
Or at least
okay.
not
I
the
don't
full name
recall.
of it. Yeah.
Well, you know what it is. If it's PEP carboxylase, what is it carboxylating? In this way, biochemistry is easy. It's the only way in which biochemistry is easy.
Right.
If it's PEP carboxylase, what does carboxylase mean? It's an enzyme that does what?
Like fixes something to carbon?
Okay, it actually adds carbon to something, but that's good enough, whether you're fixing something to carbon or adding carbon to something else. So it's taking that carbon dioxide, specifically carbon dioxide, that form of carbon, and adding it to something else. And because this is PEP carboxylase, it's adding it to this compound called PEP, which stands for phosphoenol pyruvate. But if Dr. Judd didn't give you that in class, you don't have to worry about it. But PEP is this 3-carbon compound. So you have this 3-carbon compound that PEP carboxylase adds carbon dioxide to, which now makes it a 4-carbon compound, and that's why it's called C4 photosynthesis. In C3 plants, Rubisco grabs carbon dioxide, adds it to ribulose bisphosphate, which is a 5-carbon compound. That makes a 6-carbon compound, but that 6-carbon compound, Dr. Judd is keeping the count of us. That 6-carbon compound is unstable, and it breaks into 2 3-carbon compounds. And that's why it's called C3 photosynthesis. Now the question is, why do we have, does that help you kind of understand some of the key differences? Oh, well, wait, there are other differences. So once carbon fixation happens, now you have carbon dioxide latched onto these new molecules. Carbon dioxide added to ribulose bisphosphate and carbon dioxide added to PEP. What happens to those complexes in C3 and C4 photosynthesis? Let's start with C3. You have the 6-carbon compound. It breaks down into 2 3-carbon compounds, both of which are PGA, phosphoglyceraldehyde. What then happens to those PGA molecules.
Yeah, what's phase 2 of the Calvin cycle?
Why am I doing this? Dr. Judd taught this section.
I know I've reviewed this with some people, but none of them are on the Zoom call today. We broke
So
the
what
Calvin cycle into 3 phases, right? The first one is fixing carbon, and the last one is regenerating the RUBP so you can keep the cycle going, so it's ready for the next carbon dioxide molecule. What's that middle one? What's that second phase for?
Isn't the second one reduction of carbon and adding energy?
Yes, it's reduction so that you add energy and can build sugar molecules, glucose or fructose molecules, out of the 3 carbon molecules that are produced by the carbon fixation phase.
So the take-home point is that once you have that carbon dioxide fixed to ribulose bisphosphate, and then you get those 2 PGA molecules, those 2 3-carbon molecules, you can then just immediately build glucose. That's different than what happens in the C4 photosynthesis, because when in C4 photosynthesis, you add carbon dioxide to PEP to produce this 4-carbon compound, oxaloacetic acid, or oxaloacetate. That molecule, then what happens to it? Does it go straight into the Calvin cycle and start generating glucose, or do you have to do something in between?
It has to like go into the sheath cells.
Yeah, so that's how the plant basically gets carbon dioxide out of the mesophyll cells and into the bundle sheath cells. It uses oxaloacetate that's converted into malate. And I don't remember how much Dr. Judd wanted you to know about that conversion, but that's how it gets transferred out of the mesophyll cells where there's oxygen present to into the bundle sheath cells where there's no oxygen present. And then what happens to that malate?
Does it bond to RUBP there?
It doesn't bond to RUBP. It's a 4-carbon compound because it has that extra carbon dioxide sticking onto it. What do you have to do with that extra carbon dioxide? Because what bonds to RUBP? A malate molecule?
It would just be the carbon dioxide.
Yeah, so what happens when you get into the mesophyll cell is the cell strips off that carbon dioxide. Now that carbon dioxide is free to bind with RUBP using Rubisco to do that binding. And then you just go through the Calvin cycle just like a C3 plant does. And so the Calvin cycle actually isn't different. What's different is that initial carbon capture that happens out in the mesophyll cells, and then the transfer out of the mesophyll cells into the bundle sheath cells. And the whole reason that plants are doing this is because the bundle sheath cells are protected from oxygen. Why do we want the bundle sheath cells to protect Rubisco from oxygen? Why does Nathan keep smiling?
Because I'm watching Joe think, and
Oh,
that's
okay.
because all the other people have their cameras off. That's okay.
Well, if Joe's thinking, Joe should start answering.
Yeah. What does Rubisco do sometimes in the presence of oxygen?
So sometimes it will bond to the oxygen instead of the carbon dioxide.
And you don't want that to happen. Why?
Because isn't it like it can't produce the sugar or it can't produce as much sugar?
It can't produce as much sugar. And so it really reduces the efficiency of photosynthesis. And so as a result of that, you have to use energy to free that oxygen from rubisco, so then you have from ribulose bisphosphate, so that you then can use that ribulose bisphosphate to make glucose. And so it ends up expending energy rather than storing energy in chemical form.
Now,
And so that's why you see. Go ahead, Nathan.
I was going to say, in class, I emphasized that moving those the malate into the bundle sheath cells and then freeing that carbon dioxide for the Calvin cycle to proceed normally in the bundle sheath cells costs energy. So you get this benefit of having the Calvin cycle proceed in the absence of oxygen, but you have to pay for that benefit by actively pumping the 4 carbon compounds into the bundle sheath cells and then stripping off that carbon dioxide again.
So C4 generally takes more energy than C3, but if you're in an area where it's really hot, C3 plants have to be constantly opening their stomata to let more CO2 in, because if CO2 levels get too low, ribulose bisphosphate gets oxygen added to it because there's not enough CO2. And then you enter photorespiration, so then you start using up more energy as a C3 plant if it's hot and dry. And then under those conditions, C4 plants are more efficient, because even though it takes a little more energy to get the malate out of the mesophyll cells and into the bundle sheath cells, it more than makes up for that, because once it's in the bundle sheath cells, there's no competition for access to rubisco by oxygen. All of the carbon dioxide that is there, it only bonds with rubisco because there is no oxygen to bond with rubisco. And so that's why in hot, dry areas, C4 plants have better photosynthetic efficiency than C3 plants do. And that's why C3 plants have better photosynthetic efficiency in cool, moist environments, because they can open their stomata whenever they want to, because it's cool and moist, and when they open their stomata, they're not going to lose so much water. Or if they do lose water, they can easily replenish it, and they get to do photosynthesis without running out of carbon dioxide, and they don't get the energy hit that a C4 plant has by having these extra steps of transport. Does that make sense?
Yeah.
Okay. So why are we not just giving you the answers? Because we haven't given you an answer yet, I don't think.
I think the question was asked before I signed on. I don't know what it was.
Oh, they wanted me to tell them the difference between, to go back over the difference between C3 and C4 photosynthesis.
Okay.
Why am I not just telling you what the differences are?
You want us to think.
Yes, and this is what we call in the educational game retrieval practice. You get those things in your brain most solidly when you practice the process of retrieving that information. Retrieval practice is an excellent way to learn things. Continue. What's your next question? Did we answer that question suitably enough for you, deeply enough for you?
Yeah.
Okay, good. All right. Proceed.
What's this?
What was the name of that process that like the electron got a lot of energy and it shot up and it was like a Z something?
You're most of the way there. Z scheme.
It's kind of is it called that because of the way the electron transport system is generally illustrated?
Yeah, the original drawing has it horizontally, and then almost all textbooks show it tipped over like an N, like a backwards N. But originally it was drawn as a Z. Do you remember what the z-scheme depicts, like what sort of the y-axis is on the z-scheme. The way I drew it anyway. It would be the horizontal axis in the original drawing, but the way we draw it and the way it's in your textbook, it's the vertical axis.
Was it just the energy?
Yeah, that process is showing the capture of light energy and then the conversion of that light energy into chemical energy as ATP and NADH. But what you're seeing is the pathway of the electrons as they flow through the light reactions. So they become excited and then they move down the transport chain as ATP is generated, and then they're excited again and move down the transport chain as NADPH is generated.
So the photon of light, the energy in the photon of light, it hits the electron, and the solar energy then gets converted into energy stored in the electron, and that's why it no longer stays on the atom that it currently was on, is because it's actually, you receive so much energy that it's stripped away from that atom altogether. So now you have this high energy electron bopping around. And have you ever put a black light on a Scorpion or one of those groovy 1970s t-shirts that like lights up, fluoresces under black light? Yeah, Joseph, how many of those t-shirts do you own, Joseph?
A couple.
So that's an uncontrolled situation where the ultraviolet light is striking molecules in the pigments of that shirt. They're getting knocked away from the atoms that they belong to, and then they just fall right back down back into the orbital that they just came out of. And when they do that, they let out some photons of light. But what happens in the electron transport system here is when that electron gets energized and gets knocked away from its nucleus, it actually gets captured by a protein molecule, one of these cytochrome molecules. And then it has all that energy, and the way that the cell harvests that energy is by slowly bleeding off that energy by transferring it in little packets of energy from one cytochrome compound to another until it's kind of finally bled all of that extra energy off of that electron. Then it gets another dose of energy and gets elevated again, and then it bleeds it off again. And at the end, you're left with this electron at the end, and it has to go somewhere. And it gets picked up by an electron receptor.
So if these electrons are being sent off on this little journey, ultimately to an electron receptor, and in the process, the energy is bled off to make these energy-carrying molecules, where does the system get a new electron from? Excuse me, a new electron from.
Did you go through this with him in class? I don't recall.
We did, yeah.
Okay, all right.
Just going to actually look at my slides right now. We've just talked about light, and before that we talked about carbon dioxide. What's the other input to photosynthesis?
The water?
Yeah, what's the water for?
Did it use the hydrogen, or was it, I don't remember if it was the hydrogen or oxygen.
Well, you know what happens to the oxygen. What happens to the oxygen in the water?
Oh, it's put off, so it uses the hydrogen.
So it does use the hydrogen, but I was asking about an electron. So it uses, the hydrogen does play a role in the whole system, but the splitting of water into the components, hydrogen and oxygen, makes an electron available for the light reactions to proceed. So the electron from the water gets moved to the reaction center where it can be excited by a photon of light, and it can move on down the transport chain. But then it needs a new electron for the next time. So it splits water again. So it continually splits water in order to gain that electron to be excited by light. Let's see, where did I have that? Yeah, right here. We stepped through it on the October 30th slides.
Do you want to briefly share your screen so you can show them what you're looking at?
Yeah.
We would normally, if we were doing this in a classroom up at school, we would normally have our computers with us and we could just project these things up on the screen in the room.
Right. So this antenna complex, this little yellow pyramid, that's all kinds of pigment molecules that are, you can imagine them as big receivers for photons of light. And they shuttle those, the light excites the electrons, and those excited electrons shuttle the energy to the reaction center where an electron is excited. So we're moving up this energy state with the excitement of the electron at this reaction center. Let me get rid of this. Then as the electron moves down the electron transport chain through these different cytochromes to photosystem 2, it can be excited again by another antenna complex. As this electron is going, it's going to be replaced by another electron that's also ready to be excited by the next incoming packet of energy. So electrons are stripped from the water molecule. Oxygen, gaseous oxygen is released, some hydrogen ions are made available, and they flow around in the stroma of the chloroplast. They're used for various purposes that we didn't go into detail on, and the oxygen is released and the electron goes here. Down this electron transport chain, my animations seem to be in an odd order. The energy is kind of bled off to make ATP, as we just talked about. Back here in photosystem 1, the electron is re-excited by the next photon of light. So you're up here. And then you move down another electron transport chain to synthesize NADPH, and the electron ends up with an electron acceptor. Sometimes it's confusing. Photosystem 2 comes first and photosystem 2 comes second. That's just an accident of the way we worked all this out. Well, not we, other scientists before me worked all this out. We learned how photosystem 1 works before we learned how photosystem 2 works. So we call them 1 and 2 because of the order of discovery. But this is the key. And the reason I wanted to hit this is it's a common misconception that the oxygen released by photosynthesis comes from CO2 molecules. It does not. The CO2 molecules are all interacting over there in the stroma, in the Calvin cycle. The water is important in the light reactions, which are happening on the thylakoid membranes in the chloroplast.
And so the electron receptor here is NADP, and the electrons are used to add those hydrogens to the NADP molecule. And that's a different oxygen receptor than what we have in cellular respiration, in which case that's actually oxygen, it's the oxygen receptor that, or the electron receptor there. So in this case, it's NADP.
Yeah, thanks for clarifying that. I said that a little funny before, that you can see 2 electrons going into 2 NADPH molecules.
Mm-hmm. All right. What other questions do you have?
What types of things about like the animal and grasses of the day would you like us to have known?
So what we'll oftentimes do with those things is we'll use those things in examples. So I did the section on resource limited population growth. So resource limited population growth, not resource limited population growth, but resource-based competition models. So one of the ways in which we might do this is we might say, think about Andropogon gerardii and lead plant, whose name, why can I not remember lead
Girardi.
plant? No,
Oh.
Andropogon gerardii, lead plant's scientific name.
Amorphicanescens.
Amorphicanescens, thank you.
I
I got it sitting out in my yard and I can't even remember its name. Anyway, so we have these two plants. We have big blue stem and we have lead plant. One of those is, how would we talk about in scientific terms, how would we talk about big blue stem? Like what kind of plant is it? What kind of photosystem does it use? What photosynthetic system does it use?
Isn't Big Blue Stem C4? C4 grass?
It's a C4 and it's also is it a grass or a forb?
It's a grass, I think.
Yeah, so it's a C4 grass, a warm season grass if you want to call it that, but it's a C4 grass. And Amorphicanescens, the lead plant, what type of plant is that? Is it a grass or a forb?
Forb?
It's a forb. Does it do C3 or C4 photosynthesis?
C3.
C3. So you have these two plants. They differ. What else is special about lead plant? Other than it's a really pretty plant with really pretty leaves and really nice inflorescences.
Doesn't it fix nitrogen?
It does fix nitrogen. So it's a legume. So it's in which family?
Fabaceae?
It's, what did you say?
Fabaceae?
The Fabaceae, yep, that's the family of the legumes. So if I were to, oh, I must have closed that. Hold on, let me go find something real quick. Let me share my screen with you for just a minute. So think of these two plants. If I showed you this example of two zero net growth isocyclines for two plant species, and I told you one of these was Andropogon gerardii, and I told you that the other one was Amorphicanescens, which one would be which? Now, in order to answer that question, you have to know those things that we just talked about. And you have to know how Tillman's model of resource-limited competition, resource-based competition works. So which of these would be the legume and which of these would be the C4 grass?
Would the dotted line be the C4 grass?
So the dotted line needs a lot of what?
Oh, sorry, I meant the dark line.
Okay, explain why.
Well, because it needs less water so it can do better with photosynthesis in drier climates.
Okay, so you would say that that's a C4 grass because it needs less water. What else does it need a lot of?
It needs a lot of the nitrogen.
Because it can't do what?
It can't fix it.
It can't fix it. What about the dotted line? So you're saying that the dotted line would be lead plant, right?
So it can fix it, so it wouldn't need as much of the nitrogen.
Yep, but it's a C3 plant, so it needs
More water.
There you have it. Good. So what we'll typically do with these plants and animals is that we will incorporate them into the context of other kinds of questions. And once again, what we're trying to do is we're trying to get you to have a sort of integrated view of how Kanza works as an ecosystem. And so in the same way that today when we were talking about disturbance, and I told you, when you think about why fire has a homogenizing effect on ecosystems, frequent fires in particular, and the homogenizing effect is that it gets rid of a lot of the forbs and leaves behind all of these grasses. Well, it's easiest to think about that when you think about kind of what grasses are like and what forbs are like in terms of how they grow. Once you think about that, you know where meristems are located on grasses, you know where meristems are located on forbs, and you can imagine that when a fire goes through there, it's going to be much more damaging to forbs than it is to grasses, because grass meristems are protected down there at ground level and forb meristems are up top. Once again, you're integrating a lot of information about plants into this understanding of how one particular type of disturbance will affect species diversity. And not only species diversity, but also what those species are that thrive under repeated fire regimes. So that's how we'll use it, Michaela. Does that make sense?
Yes, thank you.
Sure. Seldom will we say, Schizocosa ocreata, which of the following of these statements is true about Schizocosa ocreata? We won't typically do that, but what we might do is we might then ask you, and I've never asked this question on a test before, but we might say, the Veen et al. Paper looked a lot at how bison affect heterogeneity. There's this spider that lives on the prairie that is known to, in areas where it occurs at high abundance, we talked about this when we went through it, it's known to change the distribution in space of grasshoppers. What effect would you expect that to have on heterogeneity of plant communities on Kanza prairie? And so, because you probably didn't remember all of the details of that particular species, I'll probably remind you of some of those in the question itself, but then you have to think about that species in terms of how it might affect heterogeneity in similar ways or in different ways from, for example, bison, as an example.
You all have also done a pretty good job over this review session and in previous discussions in class and in office hours, recalling family names and knowing what's important about certain families. Grasses are in a family of plants in which C4 photosynthesis has evolved. Fabaceae is a family of plants in which symbiotic relationships with nitrogen-fixing bacteria have evolved. Those are things about those families we might want you to know, and the species we've talked about are examples from those families that are important to know about.
Yeah, and once again, knowing those things about those species tells you about what their role will be at Kanza. And that's kind of the goal here.
Good, yeah, good point.
What else? Nathan, you look like Seth Meyers at the beginning of shutdown when he was stuck in his attic.
Yeah, I have this built-in desk that I can't move, and it just backs up to a silly closet. If I had the next upgraded computer, I'd have a great virtual background. I'd probably pick some sort of a landscaping company or something.
I'm expecting Ethan Hawke to come out of the closet today.
Oh, no.
I have a quick question.
Yeah.
So I got my ANOVA back, and I was wondering if you could talk through doing SS among, because I didn't know that I did it wrong, but I guess I did it wrong.
Yeah. So the sums of squares among And keep in mind, I'm not gonna require you to calculate this because it's just gonna take too long on an exam. But I'll tell you what it is. Most people, when they did the sums of squares among, they did the sample mean minus the grand mean and squared it, but then they didn't weight it by sample size. So you have to take that squared deviation from the mean and multiply it times the sample size. The other thing that people messed up a lot that's similar to that is when they were calculating the grand mean, one of the most common mistakes I saw was people adding up all of the means and dividing by 4. But you can't do that because each mean was made up of a different sample size. So you have to weight that mean by the sample size, add up all of those weighted sums, and then divide by the entire sample size. So I think I'd have to go back and look at it, but I think the mistake that you made was not weighting the sum of the squared deviation by the sample size.
That's
And I
what
think
you.
maybe on yours, I might have actually written in a little red in your equation, but I don't recall.
You did write has to be weighted by sample size next to my
Oh, yeah.
grand mean calculation. So
Oh, well, then the same thing is also true of some squares among. If I didn't write it in there, I should have. But that's generally what some people did, which I thought was weird, is they calculated the sums of squared deviations and then they added all those up and multiplied that times 37, which also isn't the same thing as doing each one by its own sample size. It gives you a different number. So if you go to, I think I still have the ANOVA one open from earlier today when I was going through this with somebody. I'll go to the part where this is in the ANOVA PowerPoint. It's right here. And what you see is that it's the mean minus the grand mean squared times the sample size summed across everything. So you do the multiplication at the level of the mean time minus the grand mean squared. You don't do the mean minus the grand mean squared, add all those up, and then multiply that times total sample size. It's by each individual sample size. So it's a minor thing. And if it was a minor thing, I generally didn't take a ton of points off. But if you had lots of minor things that then tended to accumulate, then things got worse.
What time does it close?
I have no idea what time it closes.
Sorry.
What else can we do for you? So Joseph, on the ANOVA for the test, what I want you to be able to do is I want you to be able to look at the ANOVA table and discern certain things about the ANOVA table. So if you see the degrees of freedom for the treatment, how many treatment levels did you have? If you see the degrees of freedom for the within sample variation or residual variation combined with the degrees of freedom of the treatments, can you tell me what the sample size was? Do you know if I gave you the sums of squares, the degrees of freedom, could you calculate the mean squared and the F value for that, for example? So interpreting and working with an ANOVA table is something that you can do in the time frame that you have on an exam, whereas you can't really construct an entire ANOVA on an exam. The other thing I'll ask you to do on that front is to look at graphs that have means and confidence intervals graphed and be able to interpret those correctly in terms of which means are different from which other means and things like that. Does that help clarify what you kind of need to concentrate on for studying?
yeah, for now.
And I've said that, well, I said that earlier today, but I said that in class and I've said it repeatedly in the past as well, but it's just as true now as it was then. And if we ever do something to you on a test that we told you we wouldn't do, you can totally bring that up and we'll do something about that.
Can we maybe work on interpreting one of those tables, like go back over that?
Yeah, sure. So I'll just share my screen again in that same PowerPoint. Let's see. Okay. Yeah, so this was this, can you guys see the sugar pea growth example? Do you guys see my screen? Okay, all right, good. So we have these 5 treatments. So when we construct an ANOVA table out of this, so we basically, this is the data set. In this case, it's made up of 5 treatments. Each treatment has 10 observations. But the way that ends up working out in an ANOVA table is, so here is the ANOVA table that comes from all of these calculations. We have 4 degrees of freedom. Well, we know when we see that there are 4 degrees of freedom that we have 5 treatments, because the degrees of freedom is 1 minus the total number of treatments that we had. We have 45 degrees of freedom for within sample variation, because the number of degrees of freedom here is the total sample size minus the number of treatments that you have. So that's 50 observations minus the 5 treatments, that's 45. These two things add up to 49, which is 1 minus the total number of samples that you have in the entire experiment. So knowing about the degrees of freedom tells you things about the experiment. When I see an ANOVA table in a published article, which quite frankly, you don't see ANOVA tables in published articles anymore, which irritates the crap out of me, because there have been times when I've looked at the ANOVA table for an article, and after having read about the experiment, I know that they did something wrong with their statistics because their degrees of freedom are wrong. And so degrees of freedom are very important to put out there, because it gives the reader knowledge about the structure of the experiment. The mean squared is just the sum of the squared divided by the degrees of freedom. So you can do the mean square for among, the mean square within is just 245.5 divided by 45, and 1077.32 divided by 4. That gives you the 2 mean squared values. The mean squared among divided by mean squared within is F, and then F is what you actually evaluate to calculate the p-value. And this is a humongously big F statistic, which is why your p-value is so small, which means that there's definitely something different in this data set. And when you go back and look at the data, it should be clear to you that the something that is different is that the mean of the control is different from all the other means. The mean of the 2% sucrose is different from these means, and these means really aren't different from one another. So there are a couple of differences, and a couple of differences that you see are reasonably large, which is why then the F statistic is so large. And when the F statistic is really large, that usually means that the p-value is small. And that tells you that if you say that two things are different in that data set, your chances of being wrong are really, really low, because they are different. Does that answer your question? And this is just the R version of it. This is different from everybody else. This is different from everybody else, and these aren't really different from one another. But that thing that I just did, looking at the graph and interpreting those means in 95% confidence intervals, expect to be asked to do something like that on the test.
Are you good for me to go ahead and post the review sheet as soon as we're done here?
Yeah, if you've added all the things you needed to add to it.
Yeah.
Yeah, do it.
Let's see.
Can we go back over how to do the coexistence thing on the graph for competition?
Yeah, sure. And normally I would actually send one of you to the board and make you do this and make your friends help you through it. But since we're not in a room with a board, that's going to be a little more difficult.
Is anyone on an iPad and want to volunteer to draw as we talk through it?
Oh yeah, sure, we can do that. Volunteers? It's good for you. Well, I know what the problem is. I'm looking in the wrong file, looking for the wrong name.
Uh-huh.
All right. Now that I've got the right figure up, okay. Let me go and share my screen. Okay. All right. So okay, so sometimes it's not sharing. I've really been having trouble sharing screen lately. All right, so in this situation, we have one resource axis, which I always just use light, because light's easy to think about. And the other reason that I use light, what's that,
It's
Nathan?
also the one in the Konza Prairie case study. So.
Yeah, and the other reason is it's the one that's in VINA-OWL. So when a bison comes by and eats up a bunch of big blue stem, which can be a very, very tall grass, when it eats down that big blue stem, all of a sudden there's just more light available to any plant that was being shaded out by big blue stem. And so light can vary quite a lot in a plant community. And we normally think of light being limited in forests, but it turns out that a prairie is just like a forest. It's just a forest that's 9 feet tall rather than a forest that's hundreds of feet tall. So light can be really in low abundance or high abundance. Same thing with something like nitrogen, which is the other thing that they were talking about in terms of resources in the VINA-OWL paper. And for all the reasons we talked about today, nitrogen can be limiting because it leaches out of soils, bacteria denitrify it and blow it back to the atmosphere. It's hard to fix to begin with, so there's limitations on fixation, all kinds of reasons why nitrogen is hard to come by. And so we have these two species. One species needs a lot of light, but needs relatively little nitrogen. And so its zero net growth isocline looks like this. And on this side of this zero net growth isocline, what's happening in terms of population growth? Over here.
It'd be increasing.
Okay, population growth is increasing. And to the left and underneath this zero net growth isocline, what's happening? Over here and under here. Over here in terms of light supply, and under here in terms of nitrogen supply.
It's decreasing.
It's decreasing, and what's happening on the line?
Does it just stay the same?
It just stays the same, zero net growth. That's why it says zero net growth isocline. So would this be an early successional species or a late successional species?
Early?
Why, Alexis?
Because it needs a lot of light. So that's like after disturbances when there's going to be fewer plants to shade it.
Yep, and after a disturbance, what also oftentimes happens to nutrient supply?
like the nitrogen would have been burned off.
Burned off or leached out or both, sure. And so because you have a organism that needs a lot of light and doesn't really need any nitrogen, oftentimes you find nutrient limitation in places with either poor quality soils or really thin soils, because there's not been enough successional time for soils to build up and build up that nutrient supply. So this would probably be an early successional species, especially if you compare it to the other species in the scenario that needs very little light but requires more nitrogen. And so here, once again, what's happening to the upper right for this orange species?
It increases.
Joseph? Yeah, so positive population growth over here, negative population growth when you get either below its light limit or below its nitrogen limit. So negative population growth and zero population growth all along the line. Well, if this is what the species needs, then you would assume that this is probably reflective of what the species uses. And so we can create these resource utilization vectors. So because this species needs a lot of light and needs very little nitrogen, for every unit of nitrogen that it uses, it's going to use a lot more light. And so this triangle, this right triangle here, represents a little bit of nitrogen use relative to a lot of light use. Same thing here, this 90-degree triangle right here represents a species that's going to use very little light relative to the amount of nitrogen that is going to use. So these vectors are taken generally as indicating how much of the two resources that species is going to use based on what its needs are. So the zero net growth isocline represents what the needs are, and this vector that goes from the intersection of those two needs back to the origin represents then how fast they're going to utilize those two resources. And the way we can...
I want to ask to them, how does the ratio at which those two resources are used relate back to the line itself on the graph? What do they describe about that line?
Which line, the dotted line?
The dotted lines. Yeah. So the plants are using those two resources at their respective ratios. What are those ratios telling you about the line itself? In the case of the green one, the height of the triangle, the rise of the triangle, is the nitrogen use, and the run of the triangle here is the light use.
And back in the old days, we used to teach this particular mathematical concept by saying it's the rise divided by run. You guys ever hear that in math class sometime?
So then what is it, Joe?
Unmute yourself.
The slope?
Yeah.
Are you
Yeah,
serious?
it's a slope. And if you have a low slope,
I'm always
It-
serious.
Oh,
if you have a low slope, it means that you're using not much nitrogen relative to light. And if you have a high slope, it means you're using a lot of nitrogen relative to light. So to get to, I think it was Alexis's question, the way we figure out where species can coexist is we basically just take these two lines where they are, and rather than having them intersect at the origin, we have them intersect at the point of coexistence. And this then represents a range of resource availability during which, if you start anywhere in this resource space, because remember, we're graphing resource 2 versus resource 1. So this is different ratios of nitrogen to light. This would represent a lot of nitrogen, a lot of light. This would represent a lot of nitrogen and very little light. This would represent a lot of light and very little nitrogen. Well, you can see if you start over here, the orange species is not going to prevail because it already is not getting enough nitrogen to survive. So the green species will be the one that wins the competitive interaction. But this area in between becomes a little sketchier, but the way that you can figure this out is just by taking these resource vectors, putting them in here, and any place inside this triangle where you begin, those two species will end up at this coexistence point. But as soon as you cross this line over into here, you're too close to the limits of the green species such that the orange species will end up winning. And when you cross this dotted line over here of the green species, you're too close to the lower limits of the orange species such that the green species will end up winning the competitive interaction. So if you're in here, they'll end up at this coexistence point. If you just cross this line to a little bit, you'll end up with the orange species prevailing. You cross the green line, the green species prevails.
Be sure you understand that when you start out in that triangular region in the upper right, the populations are increasing. So what's happening to resource availability in general?
It's going down.
Yeah, that's why
Yeah.
you converge on this point of coexistence, because you're drawing down the resources.
Yeah, and when I first introduced this, I tried to get across to you how to translate this graph back into thinking about population size, and that's where Dr. Judd is coming from. If you're out here, chances are there are a lot of resources because there aren't very many individuals. And as you grow in population size, more and more individuals are going to be using these resources, and so you totally draw those resources down until you finally get to the point that there are so few resources that you can't grow anymore. You're not declining, but you're not growing anymore. And presumably at that point you would reach equilibrium between these two
It
species.
should feel a little bit like the inverse of approaching carrying capacity as you use up resources.
Yep. Does that help, Alexis?
Yeah.
That was a combination of me making you give me answers and me giving you answers. What else can we do for you?
I have to go, but thank you guys for doing this. I appreciate it.
Sure, no problem. Hopefully it was helpful to you.
I know we went over it in class like all day, but what about the Kaza Prairie should we, like what about, not the Kaza Prairie, but Veen et al., what specifically about this study should we know?
Well, you should certainly know the ways in which fire and grazing are different from one another as disturbances, just kind of in their very nature. But then you should be able to then translate those differences into the reasons why Veen et al. Expected fire to have one effect on species diversity and they expected grazing to have a different effect on species diversity. And also they expected fire and grazing to have different effects on heterogeneity. All go back to kind of the fundamental differences in terms of what fire does as a disturbance versus what grazing by bison does as a disturbance. So you should certainly know that. You should know what their experimental design was like. The 1 year burn versus 4 year burn, the grazing versus non-grazing. You should know what their hypotheses were about. What was the hypothesis about what the effect of fire should be? What was the hypothesis about what the effect of grazing should be? What was the hypothesis about how those two things interact should be? You should also know, go ahead, Nathan.
I was going to say you should be sure you understand the graphs that were in the paper. You should be able to read them and interpret them. And, and you should know what different things they measured, right? We've talked a lot about diversity and heterogeneity and things like that, but we've also talked about how those things are measured. So that goes back to the whole lecture we had at the beginning of this unit on
Whole 2
measuring
lectures.
diversity.
Yeah, we spent 2 days on that.
Yeah.
A number of questions that I had from people when they were coming in to ask questions about writing their Veen et al. Summary was, how do we calculate diversity? And I was like, Dr. Judd went through that with you in class. And so certainly go back and review how we quantify diversity and get straight in your head what species evenness is, what species richness is, what species dominance is, and how those things all relate to diversity. For 2 reasons, it helps you understand the Veen et al. Paper better. But then the other reason is that Dr. Judd went through that with you and had you actually do a homework assignment where you were calculating those things.
The 4th component to diversity that we spent time on in various office hours, but also we spent time on today in class, was heterogeneity. And that relates directly to what we talked about in the lecture, species composition. So when we're not worried about exactly who the species are, we can do richness, dominance, and evenness. But once the identity of the species matters, then we look at composition and we can measure things like heterogeneity in the plant community.
Yeah, because you let me pull something up once again that I was using to answer somebody else's question. Oh, where is it? Ah, there it is.
There were several times in different office hours, Joe, I don't know if you were one of these, but there were several students that I've met with where we talked a little bit about composition and heterogeneity, but then to really make sure we understood it, we actually worked through and interpreted the equation for percent similarity in the Veen et al. Paper, which maybe is a little overkill, but it guarantees that you understand what it is we're talking about.
So this is something that I put together. Michaela is not here, but she sent me an email asking about heterogeneity. Can you guys see these 2 blocks?
Mm-hmm.
So imagine that this is a sampling unit on Kanza. Imagine that this is another sampling unit on Kanza. And these numbers actually just refer to the number of species that are there. So it's just species richness. Well, when you look at the one on the left, you see that the individual subplots within the sampling area are all very similar in terms of the number of species that you have there. On the other hand, the one on the right is very different in the number of species that you have there, such that just on the basis of species richness, which of these would be more heterogeneous?
Probably the second one.
Yeah, the one on the right would be more heterogeneous from the perspective of number of species. However, if these 12 species in this sampling plot and these 13 species in this sampling plot were all different species, even though they have a similar number of species, they would be very heterogeneous in terms of species composition because none of these 12 species match any of these 13 species. So heterogeneity can be defined differently depending on what kind of measure you're asking is heterogeneous. So these things could be just as heterogeneous as these things if many of these species are actually different from plot to plot. If these 12 species and these 13 species, 11 of the species are actually shared between them, they're heterogeneous not only because they have similar numbers of species, but they're homogeneous because they have similar numbers of species, but they're also homogeneous because those species are the same. And so Michaela was having difficulties kind of separating diversity from heterogeneity. And so this would hopefully help with that.
Where composition and species identity play a role.
Yep. Brandon, you have been very quiet. What are you lurking about?
I came in here and I didn't really have any questions, but I was just wondering if I learned anything I needed to know for the test.
Well, if you have any questions, get them out there.
All right.
I think I've asked about everything I need to ask
Okay.
until I start working, and then
Okay.
we'll
Well,
see.
over the weekend, if you have questions, feel free to drop us an email. We're generally on email off and on throughout the weekend. We won't necessarily respond immediately, but we'll see it and get back to you.
Sounds good. Thank you, guys.
Alexis, do you have anything else?
I think I'm done.
All right, the
Thanks.
study guide's posted.
All right. And Brandon?
I'm good. Thank you.
All right. Then I guess we're done.
All right.
All right. Good luck studying this weekend.
Have a good evening.
Thank you.